Okay, doctor.
On the left side, you can see all slides and the main slide. Yeah, okay, I don't know what the problem. Okay, let's go on. On this slide, you can see correlation. So results of scientific research, demonstration, correlation between maternal age and incidence of chromosomal abnormalities. And these chromosomal abnormalities were detected using chorionucleus sampling and amniocentesis. So on this slide, you can see that after maternal age 36 years old, the incidence of chromosomal abnormalities is increasing extremely. Can you see?
Yes, madam.
Especially after 44 years old. Next question we'll discuss will be general characteristics of chromosomal diseases. First one, chromosomal diseases arise in gametes or at the first divisions of zygote. Second one, characteristics, all states of pathogenesis fall at the prenatal period. That's why it's very difficult for us to study pathogenesis of chromosomal abnormalities. So, multiple congenital malformations are the main clinical features of chromosomal diseases. The etiological and pathogenetic therapy is not developed for chromosomal diseases. Unfortunately, we can use only symptomatic approaches for the treatment. And last one, characteristics. It is possible to apply prenatal diagnostics and using of prenatal diagnostic methods, including non-invasive and invasive prenatal diagnostic. It's one of the basic approaches for the diagnostic of chromosomal diseases. We use cytogenetic method. Cytogenetic method cytogenetic method can help us with diagnostic of chromosomal diseases because we should to calculate number of chromosomes in a karyotype of the fetus and to analyze presence of or absence of different structural chromosomal abnormalities. So, doctors, can you see slide number 12? Classification of chromosomal mutations.
Yes, madam.
Okay, good. So, all chromosomal mutations may be divided on the following groups. First one, it's numerical abnormalities of chromosomes. Second one, it's structural abnormalities of chromosomes and different cell lines. Mixoploidy associated with mosaic forms of chromosomal diseases. So, numerical abnormalities may be divided on polyploidy, including triploidy, tetraploidy. Polyploidy is additional set of chromosomes associated with haploid set of chromosomes. So diploid set of chromosomes will be 46, haploid will be 23. So presence of 46 plus 23 plus 23 plus 23 and so on, it will be an examples of polyploidy. Aneuploidy is presence of absent of 1, 2, 3, and more chromosomes, not associated with a haploid set of chromosomes. Structural abnormalities include translocations, reciprocal and Robertsonian, and deletions and insertions and inversions, inversions may be divided on paracentric and pericentric versions, and rings such as isochromosomes. If we can see in karyotype different cell lines, including normal set of chromosomes and abnormal set of chromosomes, it will be associated with mosaic forms of chromosomal pathology. So, numerical abnormalities of chromosomes. Numerical abnormalities involve the loss or gain of one or more chromosomes, which is referred to as aneuploidy, or the addition of one or more complete haploid complements, which is known as polyploidy. Loss of a single chromosome results in monosomy. Gain is monosomy. Gain of one or two homologous chromosomes is referred to as trisomy and tetrasomy, respectively. Trisomy, its presence of an extra chromosome, is referred to as trisomy. Most cases of Down syndrome are due to the presence of an additional chromosome number 21. So we call it also trisomy 21. Other autosomal trisomies, which are compatible with cerebral to term, are Patau syndrome, trisomy 13, Edward syndrome, trisomy 18. Most other autosomal trisomies results in early pregnancy loss with trisomy 16 in a particularly common findings in the first trimester spontaneous miscarriages. The presence of an additional sex chromosome X or Y has only mild phenotypic effects. Doctors, on this slide, you can see mechanisms of numerical abnormalities, formation. So segregation in meiosis of a single pair of chromosomes in a normal meiosis. You can see on the picture A. On the picture B, you can see non-disduction in meiosis 1, and on the picture C, you can see non-disduction in meiosis 2. So these problems will be associated with dysomic gametes, which will lead to trisomies. And the disjunction in meiosis 2 may be associated as with both trisomy in case of dysomic gamete and monosomy in case of presence of no somic gamete. So, and here you can see a table which can demonstrate us different variants on the presence of chromosomes in different gametes may be associated with aneuploidies such as trisomy, presence of 3 X chromosomes for an example, tetrasomy. Here you can see presence of 3 X chromosomes and 1 Y chromosomes. And monosomy, presence of 1 X chromosome or 1 Y chromosome. such as X0 and Y0, and normal variants such as XY, XX. So let's speak about numerical abnormalities. Abnormalities associated with abnormal number of chromosomes. We'll start from trisomy. Trisomy 21 is usually caused by failure of separation of one of the pairs of homologous chromosomes during anaphase of maternal meiosis I. This failure of the bivalent to separate is called non-disjunction. Less often, trisomy can be caused by non-disjunction occurring during meiosis II when a pair of sister chromatids fail to separate. Either way, the gamete receives two homologous chromosomes, disomy, and its subsequent fertilization occurs as trisomy conceptus results. The origin of non-disjunction. The consequences of non-disjunction in meiosis I and meiosis II differ in the chromosomes found in the gamete. An error in meiosis I leads to the gamete containing both homologous of one chromosome pair. In contrast, non-disjunction in meiosis II results in the gamete receiving two copies of one of the homologous of the chromosome pair. Here you can see on the slide, so the origin of non-disjunction. Parental origin of meiosis errors leading to aneuploidy, and maternal. So on this slide, you can see that most cases of different chromosomal abnormalities, including numerical abnormalities, will have maternal cause. Only monosomy on X chromosomes, Turner syndrome, in most cases will depend on paternal reasons, and as for polysommy on X, polysommy on sex chromosome, like 47XYY, additional Y chromosome will be depend on paternal reason or paternal cause. As for trisomy 13, 18, and 21, so you can see maternal origin and polysomy on X chromosomes, or syndrome triple X also will be associated with maternal reasons. Non-disjunction can also occur during an early mitotic divisions of the developing zygote. This results in the presence of two or more different cell lines, a phenomenon known as mosaicism. So mosaicism is presence of two different cell lines, two or more, with normal karyotype and abnormal karyotype. The frequency of mosaic form is around 1% among all cases of chromosomal pathology. And we know that clinical picture in mosaic forms will be not so expressive. So monosomy is the absence of a single chromosome is referred to an monosomy. So monosomy for an autosome is almost always incompatible with survival to term. Lack of contribution of an X or an Y chromosome results in a 45X X0 karyotype, which causes the condition known as Turner syndrome. As with trisomy, monosomy can result from non-disjunction in meiosis. If one gamete receives 2 copies of a homologous chromosome, disomy, the other corresponding daughter gamete will have no copy of the same chromosome, so-called nullisomy. Monosomy can also be caused by loss of a chromosome as it moves to the pole of the cell during anaphase, an event known as anaphase lag. So polyploid cells contain multiple of the haploid number of chromosomes, such as 69 triploidy or 92 tetraploidy. In humans, triploidy is found relatively often in maternal growth from spontaneous miscarriages, but survival beyond mid-pregnancy is rare. Only a few triploid live births have been described, and all have died soon after the birth. Triploidy can be caused by failure of maturation meiotic divisions in an ovum or sperm, leading, for an example, to the retention of a polar body, or to the formation of diploid sperm. Alternatively, it can be caused by fertilization of an ovum by 2 sperm. This is known as dyspermy. Triploidy usually results in early spontaneous miscarriages. Structural abnormalities of chromosomes. Structural chromosomal abnormalities result from chromosome breakage with subsequent reunion in a different configuration. They can be balanced or unbalanced. In balanced translocation, the chromosome complement is complete with no loss or gain of genetic material. Consequently, balanced rearrangement are generally harmless, with the exception of rare cases in which one of the breakpoint damages an important functional gene. However, carriers of balanced translocations are often at risk of producing children with an unbalanced chromosomal complement. When a chromosome abnormality is unbalanced, the chromosomal complement contains an incorrect amount of chromosome material, and the clinical effects are usually very serious. A translocation refers to the transfer of genetic material from one chromosome to another. A reciprocal translocation is formed when a break occurs in each of 2 chromosomes, with the segments being exchanged to new derivative chromosomes. A Robertsonian translocation is a particular type of reciprocal translocation in which the breakpoints are located at or close to the centromeres of 2 acrocentric chromosomes. So on this slide, you can see mechanisms of Robertsonian and reciprocal translocations formation. First one, reciprocal translocation. So on this slide, you can see 2 acrocentric chromosomes. Acrocentric chromosomes will have very short arm P and long arm Q. So when 2 acrocentric chromosomes lost their short arms, and combine in large metacentric or submetacentric chromosome. At the region of centromere, it will be an example of Robertsonian translocation. We know the following pairs of acrocentric chromosomes: 13, 14, 15, 21st, and 22nd. And when they lose their short arms and combine during the centromere, it will be Robertsonian translocation. Robertsonian translocations are one of the basic mechanisms of structural chromosomal abnormalities formation. So, short arms of acrocentric chromosomes, as a rule, consist of as a rule include so-called ribosome genes. So ribosome genes are included in short arms of all acrocentric chromosomes, and losing these short arms will not have any clinical peculiarities. But carrier of Robertsonian translocation will have a very high risk of chromosomal abnormalities for their offspring. So here on the other picture, you can see an example of reciprocal translocation. This translocation involves breakage of at least 2 chromosomes with exchange of the fragments. So fragments from chromosome A transferred to chromosome B and fragment from chromosome B will transfer to chromosome A. Next one, structural abnormality of chromosome is deletion. Deletion involves loss of part of chromosome and results in monosomy for that segment of the chromosome. Usually, a very large deletion will be incompatible with survival to term, and as a general rule, any deletions resulting in loss of more than 2% of the total haploid genome will have a lethal outcome. So an insertion or duplication occurs when a segment of one chromosome becomes inserted into another chromosome. If the inserted material has moved from elsewhere in another chromosome, that the karyotype is balanced. Otherwise, an insertion causes an unbalanced chromosomal complement. Inversion. An inversion is a two-break rearrangement involved a single chromosome in which a segment is reversed in position. So in this segment will be inverted. If the inversion segment involves the centromere, it is termed a pericentric inversion. If it involves only one arm of the chromosome, it is known as paracentric inversion. So on the slide, you can see mechanisms of pericentric when centromere will be not involved in this inversion, and only long arms of chromosomes will be involved in this inversion. So short characteristics of chromosomal syndromes. Down syndrome is a condition derives its name from Down, who was the first to describe this syndrome in the clinical lecture reports of the London Hospital in 1866. The chromosomal basis of Down syndrome was not established until 1959, but Later, in Paris, it was described. So, most cases of Down syndrome are due to non-disjunction of chromosome 21 arising during the first meiotic cell division in oogenesis. A small number of cases arise in meiosis II during oogenesis, during spermatogenesis, or during mitotic cell divisions in the early zygote. So on the slide, you can see three main cytogenetic types of Down syndrome. First will be simple trisomy associated with additional 21st chromosome, and karyotype will be 47XX or XY21+. When we speak about karyotype, first of all, you can see number of chromosomes in karyotype all in all. After that, sex chromosomes, and after that, additional chromosome. For an example, 21+. So this form is not inherited. Second form of Down syndrome is translocation form, and here you can see an example of this form. This form is inherited. And last one, it will be mosaic form. Mosaic form, it's presence of two different karyotypes, and we karyotyped with normal set of chromosomes and abnormal set of chromosomes. First of all, you can see here abnormal karyotype, and after slash you can see normal karyotype. This form also is not inherited. Risk of inherited translocation from mother, father, and if a translocation in both parents occur, risk will be 100% risk of chromosomal pathology for the offspring. Down syndrome is one of the most frequent syndrome, chromosomal syndrome, and the incidence is approximately 1 in 650 to 1 in 700. So, risk factors of, excuse me. What the problem? Doctors, can you see a demonstration of the screen or not?
Yes, doctor.
Yes, okay, good. Risk factors for Down syndrome are maternal age. Maternal age. On the previous slide, you could see that after the 35, 36 years old, the incidence of Down syndrome will be increasing. And on this slide, you can see correlation between age, maternal age, and incidence of Down syndrome. You can see, for an example, 20 years old. The incidence will be 1 in 1,500, and for an example, in 45 years old, 1 in 30. So, common clinical picture of Down syndrome will be include hypertonia, sleepy, excess nuchal skin, brachycephaly, epicantic folds, protruding tongue, small ass, upward sloping palpebrous fissures, single palmine crease, small middle fangs of fifth finger, wide gap between first and second toes, atrial and ventricular septal defects, common atrioventricular canal, patent ductus anteriosus, and others such as anal-atresia, duodenal-atresia, short stature strabismus, and so on. Affected children show a broad range of IQ between 25 to 75. Social skills are relatively with advanced, and most children with Down syndrome are happy and very affectionate. Adult height is usually around 150 centimeters, and in the absence of a severe cardiac malformations, which lead to early death in 50–20% of cases, average life expectancy is 50–60 years. Most affected adults develop Alzheimer's disease in later life. So here you can see an example of epicanthus, flat bridge, a flat face, protruding tongue, epicanthus also, excess nuchal skin, dysplastic ears, brachydactyly, the single palmar crease. The hands of the adults with Down syndrome. Note that the single palmar crease in the left hand plus bilateral short curved fingers, narrow palate, abnormal shape of teeth, geographic tongue, mental retardation. Doctors, now we'll have a break 10 minutes. Can you hear me, doctors?
Yes, madam. Thank you.
Yes, madam.
Okay. Information for group leaders. Please send me by mail a list of absent students. Okay. By mail. Can you hear me, group leaders? Doctors?
Yes, madam.
Yes, madam.
Okay, I will repeat later this information. Please send me by mail. In the topic of the mail, please write number of your group. Number of your group. For an example, group 25. And list absent students. Okay.
Okay, doctor.
We'll go on in 10 minutes.